2 Wochen, 10 Tage später. Ja. Das ist das Wichtigste für die Gärtner, nicht? Ist, ob es über Nacht friert. Ja. Also bei uns warten sie noch eine Woche. Jetzt. Ja, dein Bruder ist in Montreal. Was macht er dort? Okay. Er ist ein Arzt, oder?
Nein, er ist Genetiker.
Okay. Okay.
Aber wirkt auf die Virusdiagnostizierung.
Okay.
Ja.
Wenn wir Zeit haben nachher, dann gebe ich dir Updates über die kanadische Politik und so. Ja.
Mit meinem Magen muss ich noch rausfinden.
Wohin? Ja. Können wir diskutieren. Ja. Kein Problem. Habe ich volles Verständnis. Ja, kommen schon Leute, ja? Ja, okay. Mache ich dir Platz.
Okay, good. Good morning, everybody. It's a great pleasure to welcome CHRISTOPH Simon, who I know for very many years. We shared even an office here in Vienna. CHRISTOPH did his bachelor at the University of Vienna and then his master at École Normale Supérieure in Paris, and then came back to Vienna to do his PhD with Anton Zeilinger. He was there when the locality loophole was closed, and is also on that paper. His PhD was, at that time, very fashionable topic about optical quantum cloning. But also he did many other things in quantum contextuality and standard subject of quantum foundations. After that, he went to work with Dirk Baumeister, first at the University of Oxford and then UC Santa Barbara. And then he got an offer from CNRS in 2004 for a permanent position in Grenoble. And he took this and was then on leave for 3 years to join Nicolas Gisin in Geneva in his group. And in 2009, I think he moved to Canada and University of Calgary, where he is from 2016 a professor there. His work and interest is in macroscopic quantum phenomena, also on practical quantum information in terms of quantum networks. I think today was a paper by you on quantum, on QuantPH or some resubmission.
Oh, yeah.
Okay, you don't know. I've seen this.
I'm traveling right now.
Okay, you're traveling. It was about internet and quantum networks and that type of research with repeaters and purification. And let's say most recently, but still for quite some time, he's interested in quantum biology and where the quantum mechanics plays some role in biology. And today you were going to talk about that topic. Thank you very much.
Yeah, thank you very much, Czeslaw. I have my own. Oh, okay. Can you hear me? Okay, like this, yeah. Let's see if I can make you see my slides too. That would be good. Okay, super. All right, I think we're in business. So yeah, thanks a lot for the nice introduction and for giving me the opportunity to talk about this here. This is indeed an increasing sort of part of my research. I started in this area, I would say in 2014 roughly, and then 2016 we had our first paper. But in the meantime, we have written many papers on this, and like that part of my group is now probably more than half of the group, I think. So it has really grown. All right. So topic is okay. Here you can see Calgary, beautiful city by the Bow River. There's the Rocky Mountains in the background. It's cold in the winter, but it's very nice. And here's my title, which you had a lot of time to read. Let's see if we can advance. So why? So the question was, does nature use quantum tricks somehow? So why is that a reasonable question? I would say because we know from the technology side, right, that these fundamental quantum phenomena like superposition and entanglement are unique, right? They cannot be they cannot be explained classically. They cannot be emulated classically easily. And we do know that they can provide advantages in certain applications, like there's examples in sensing, in communication, of course in computing. So, nature had a few billion years to maybe also realize that, right, and make use of them somehow. So I think it is not at all a crazy question, in my opinion. It's really quite reasonable. Nature has discovered many things and is using many physical principles that it took us a long time to understand, right? So why not these ones? That is the main starting point, I think. Then, if one accepts that, then, as Czeslaw sort of hinted at, I have spent a lot of time in my life thinking about quantum hardware, sort of how would you implement quantum technology, what's the best way of implementing certain quantum technologies in practice. So then once I had sort of convinced myself that this was a good question and we should think about this in earnest, then the question was, how would nature do it? What ingredients does it have available? What are the constraints, and like what systems could be good, under those constraints? Because obviously, most biological systems exist at like pretty high temperatures, right? Like our body temperature, 37 degrees or so. Like most animals have similar body temperatures. There's of course some that can, live at very low temperatures, but that's really not the rule. Then definitely we are condensed systems, right? Like we are not vacuum, we are not in the gas phase. Like so you have to work with these conditions, but we know also from our technology experience, we know that we can, right? Like we are aware of some systems that can have good quantum coherence and quantum entanglement, even at room or body temperature, even in condensed matter. A famous one would be, for example, the nitrogen vacancy center in Diamond, right? People have done quantum gates between electron and nuclear spins at room temperature. It works just fine. And then, so electron spins, nuclear spins should definitely be on the list, right? We also know from spin resonance experiments that these systems have quite decent, or can have quite decent coherence times in biological matter. Like you can have at least like a microsecond, maybe even 10 microsecond coherence times for electron spins. That's quite normal. You can see that in EPR. And for nuclear spins, the sky is the limit, right? Like a second is easy, but under the right conditions, it can be much longer. So it's really not hopeless just from that perspective, okay? Then the other really interesting system is photons, because we know, again, that you can send a photon through an optical fiber that is at very high temperature, it can be exposed to the sun or whatever, and the photon is just fine, right? It conserves its it's quantum information. If it's encoded in polarization, for example, you can send it for 100 kilometers, it's fine. So again, okay, not totally hopeless. Then on the other hand, though, there is some systems that I think one should probably not have too much hope for. Like, so, and one example that has been much discussed is charges, somehow superpositions of charges in different locations. That's definitely not what I would use if I was nature, right? Like we know from experience that even at low temperature, those tend to be tricky, but like at higher temperatures, for sure, coherence times tend to be femtoseconds or maybe picoseconds if you're lucky, right? So that's not what I would use. Like other people have proposed, I'm mentioning here, you know, Penrose, Hameroff ideas. Other people have proposed such biological implementations too, but I would not go there myself, you know, as a quantum engineer kind of guy. Okay, so I will focus, and my research is focused on the ones that I put in blue here, you know, which are spins of different kinds and photons. All right, so for the spins, there is an important concept that will sort of go through a lot of this talk that I want to introduce. Oh, it's covered up. I don't see it here, that window, so I don't think I can move it. Let's see. It's not the most important picture, maybe. Let's see. Maybe we can do some tricks. Let's see what happens if I go. Yeah, how about that? Is it too small? Or? I don't know how much. Inside, you mean? Mm-hmm. We could try.
But is it zoom? Or you could use the Windows key and plus to zoom. Then also the taskbar goes away.
From this view or from the other view?
From this view.
From this view, you're saying the Windows key?
Question is just whether it gets CHRISTOPH.
No, that doesn't. Oh. That sure zoomed. That may be more than we wanted.
And then while moving the mouse, you can choose which part of the screen.
Okay.
And if you just move it, yeah.
I see. Okay. All right. Okay. Getting the hang of it. Nice. All right. We'll get to that. So oh, no, it's covered up again, of course. Okay, but we can move around. So the radical pair mechanism. Okay, that's what I want to tell you about. So radicals are molecules that have unpaired electrons. Well, this is the definition. Often they are formed by one. So in molecules, like in biology, most of the time the electrons are paired up, right? They form these bonds and they pair up. So, but pretty often, you know, one might hop from one molecule to a neighboring molecule, which will then create, like if initially you had two electrons that were, oh, nice. If initially you had two electrons that were paired up sitting on this molecule over here, and now one hops over there, now you have two unpaired electrons, right? That is known as a radical pair. Since these electrons were often, initially, when they were in the same molecule, they were often in singlet states, for example, then if they hop, very often, at least for a little while, they will still be in a singlet state. Let's say now, so just the act of the electron hopping can create an entangled state now of these two spins that are, you know, pretty close to each other. If they were able to hop, it's probably a nanometer or maybe 2 nanometers or something. But now I have an entangled state between these two molecules that lives for a certain amount of time. Okay, and that time can be a microsecond or something like that. It's not a femtosecond. all right? Not necessarily, at least. And the molecules can even move around, so the distance between them can even, you know, change over time, and they can come back again, and they might react, okay, when they meet again. You can imagine maybe now the electron hops, now this molecule goes away, but comes back, and now they react again. Now, the interesting thing is that there is often, okay, if there was just there's always an external magnetic field, right? The Earth has a magnetic field, so you already have that as a background field. But that is more or less a constant field. So, as you know, a singlet state, for example, is an eigenstate of that, right? So the physicists in the room at least will know. So in any case, if you just had the constant field, then not much of interest would happen. They would just stay in the state that they were in initially, and then when they meet again, you know, maybe this one hops back or something, nothing much would happen. But in practice, often, there is other sources of magnetic fields in these molecules, namely nuclear spins. Okay, because many atoms, many biologically relevant atoms do have, for example, hydrogen, have a nuclear spin. And now you have an inhomogeneous field. You know, you have these little hydrogen spins that break this symmetry of the large applied field. And now what can happen is that as, you know, maybe these molecules move around, they are no longer just in the singlet state, let's say that they started out in, but they are going to oscillate between that state and some other triplet, we call them in physics states, and they are shown here. Okay, now I don't quite see what you guys see, which is kind of weird. How do I get out of this now again? Since you got me into it. Windows and minus. Okay, because I don't think we need it anymore. Oh no. Okay. So these are the states that I'm talking about. Okay, so the singlet state would be looks like this: up, down, minus, down, up. So these are just the spins of the two electrons that we are talking about. But then there are these other states that would be, so for the physicists, again, this is the spin 0 subspace, right? This is the spin 1 subspace. And what happens when you have, you know, these nuclear spins nearby, then the state of your electron spins can oscillate between this, let's say, initial state and then these other guys. And then, if that happens, so if they started out in a singlet state and they meet again and they are still in a singlet state, then it's easy for this electron to hop back because it sort of fits, right? Because they want to be, let's say, singlet when they are paired up and forming a double bond. But if the molecule comes back and now these guys, their spin state has evolved and they are now in a triplet state, one of those other states, then it can't go back because of the Pauli blockade. Okay, it doesn't fit anymore. So, and that is how the magnetic field, under those conditions, the magnetic field together with the hyperfine coupling to the nuclear spins can influence, therefore, which reactions happen or don't. Okay, so that's the key idea. So like if it wasn't for the nuclear spins, it wouldn't work. You know, if there was just the external magnetic field, these would still be eigenstates and the magnetic field wouldn't do anything to the reaction probabilities. But the combination of the magnetic field and the nuclear spins that are there in the molecules make it that now the magnetic field can change the outcomes of reactions, or the probability of certain, or the rates of certain reactions to happen. Okay, so this is a very like core concept in what the chemists call spin chemistry. But it is really, it's a quantum biology phenomenon. Okay, we are really talking about states that are, some of them are entangled. We are talking about, you know, hyperfine coupling. So it's very many quantum concepts are wrapped in this. You can think, that's a point I almost forgot to make. So you can think of the fact that the recombination, right, the electron hopping back, let's call that the the recombination, and I was saying that just because of the Pauli principle, it's spin selective, right? This is a spin selective thing, and if you are quantum minded, then you can view this as a partial Bell state measurement, right? If you are saying that, like, if it's in a singlet state, it can go back, and if it's not in a singlet state, it can't go back. That's, you know, this means you are projecting into the singlet subspace or the triplet subspace. It's really like a Bell state measurement, a partial Bell state measurement in that sense. So this is a thing that is a concept that's very important, and it makes it possible now for yeah, like I said here, reaction yields to depend on the magnetic field, even under conditions where the energies, like the magnetic energy, let's say the Simon energy of the spins, is many orders of magnitude smaller than the thermal energy. So usually you know, a normal chemist, if you tell them that the energy of some phenomenon that you are talking about is much less than the thermal energy, they're going to say, oh, then it doesn't matter. Okay? So this is true 99.9% of the time. But here it's not. Okay, so here there's a phenomenon because of this spin dynamics where even something that has a tiny energy has a huge impact on what's going to happen. Like even though it's much less than the thermal level. So that's, you know, this is why this is exciting. Yeah, now I'll tell you, you know, how it comes up. So first of all, the first context where it was really studied a lot is bird navigation. Okay, so the biological fact is that many birds pretty clearly use the magnetic field to find their way. You know, they have a sensorium somehow for the magnetic field. It is not clear exactly how they do it. Like so far, it's the only sense that we know, to my knowledge, that we know exists where the organ hasn't actually been identified. So people are still debating, you know, where they do it. It's probably in the eye, like in the retina, but this is still a little bit debated. So the actual, you know, real organ isn't clear, but there is a leading candidate for the mechanism by which they do it, you know, more like the chemistry and physics mechanism, and it is this. Okay, so it's the radical pair mechanism. That was the idea, at least. So that was first suggested by a German quantum chemist, quantum physicist called Klaus Schulten in the 1970s. Then by the year 2000, he had sort of narrowed it down to a particular protein, he thought, you know, so he suggested that, or a class of proteins that are called the cryptochrome proteins, and they also had like the first ideas of what exactly which radicals, but this is actually also still debated, right? So there is sort of a dominant radical that I drew over here. I'm not sure if I really want to go into the details, but so this is part of the protein, and there's so these are amino acids, tryptophan. These are different amino acids, and there's a story, you know, where there's always electron hopping, right? You need electron hopping, and these are sort of the hopping paths, and there's this FAD that's not strictly speaking part of the protein. That's a molecule that likes to interact with the protein. And the radical pair would be between this tryptophan here and this FAD here. That's sort of the main one that people are thinking about. But it's not 100% clear that this is really this is really the radical pair that the birds are using. What is clear now is that cryptochrome in vitro really is magnetic field sensitive. Like this was, for example, studied in this paper here fairly recently. However, to be honest, they need a little bit higher field, so there's still some mysteries too. Okay, they need in vitro, it needs higher fields then. So there has to be some amplification mechanism probably in biology that we still don't understand. But a lot is like sort of this is thought to be the leading explanation. You know, I think if you just survey people, they'll say that this is probably what the birds are doing, but it's not sure. Okay, but anyway. this is not our work. This is sort of came before our work. What we did is this. Okay, so this was mainly sort of in the early days with my PhD student Hadi Sadeh Hagigi. We were sort of new to this field, motivated by our own things, and we started with a kind of survey, and we realized that there's really a lot of papers on magnetic field effects in biology, far beyond just the birds, finding their way, far beyond bird navigation. There's hundreds, literally hundreds of papers where people applied a magnetic field to some biological system and saw some effect. Okay, and this goes through many areas of biology. Like reactive oxygen species are often involved, and for good reasons. I'll come to that a little bit. Calcium signaling is another big area. Stem cell development in general, the circadian clock, neurogenesis, many, many things. Okay, so this seems to be a very general thing, that biology, many processes in biology are sensitive to these magnetic fields. And it's reasonable because, hopping electrons are not rare, right, in chemistry. It's not, this is not something that's confined just to this particular one protein or just to the birds finding their way, right? But at the same time, it's hard to understand because, like I said before, there's really, this magnetic, we are talking about weak fields here, microtesla, millitesla fields. So the energies associated with those are much smaller than the thermal energies. So it almost has to be, from our perspective, that all of those things are actually quantum biological. Like, and the radical pair mechanism is a very good, already sort of prepared explanation for how this could happen. So we proposed, in this paper, I think I have another slide on it, maybe. Yeah, so then we wrote this manifesto, okay? So in 2022, we wrote this paper here, which is called Magnetic Field Effects in Biology from the Perspective of the Radical Pair Mechanism, where we started by doing a survey of all this biological literature. And again, this has 600 citations, 600 references, where we showed, how many of these things have been done. And we had already by that time written a few papers about specific cases where we showed that the radical pair mechanism could explain some of these phenomena. So typically, again, I shouldn't spend, I don't have so many slides, but okay, I should probably go a little bit faster. But anyway, so we had already looked at anesthesia, because the two main kinds of effects that people see is either they apply a magnetic field and they see some change, and for example, or maybe they remove the magnetic field. You can shield the Earth's magnetic field. Then that's called the hypomagnetic field, and you can see often pretty strong effects of that. Or sometimes what you also can do is you can play with the isotopes of a certain element. So you can, because different isotopes can have different nuclear spins, and you can see in some cases that makes a difference. Okay, and you can even, sometimes you have isotopes where you have maybe 3 of them, and this one, so they have different masses, which can also make a difference. But sometimes you can see that, I'll have an example later on, where if you have enough of them, you can really convince yourself that it's a spin effect and not a mass effect, is what I'm trying to say. So yeah, anyway, I don't want to talk too much about these because these are still just theoretical sort of proposals where we showed that we can explain what people see, with a radical pair mechanism. But now we can do better. Okay, so now what I will show you now in a lot of the rest of this talk is that we can now make successful predictions. Okay, so there's a few examples now where we said, okay, if the radical pair mechanism applies, then something specific should happen, and then somebody or us ourselves, we did the experiment and we confirmed those predictions. So I think that's a significant step forward. And it's interesting, I think, that we can do that even though we often don't know the detailed chemistry, okay? Because the predictions are really based on the physics of this radical pair mechanism. You need to set a few numbers in your, so you have a Hamiltonian, you have a master equation, you need to set a few numbers for the hyperfine couplings and the rates and so on, but you really don't necessarily need to know what molecule you are talking about, because it's a very general physics type thing, okay? That's something that the chemists sometimes have a hard time with, in my experience, at least some of them. You know, when we send our papers for a review, then the chemists will complain, you know, but you don't know the chemistry. And then our typical response is like, yeah, but we can still make predictions. That's actually kind of cool. We are hoping that you guys will help us with the chemistry, right? So it doesn't really stop us from making these predictions sometimes. All right. So now come my 3 examples, okay? So where we made successful predictions. So the first one has to do with these guys. So these are planarians. So this is Wendy Bean's group at Western Michigan University. They have been studying these worms for like a long time. They are very, very fascinating creatures. So you may have heard of the planarians. They are the guys where you can cut them in 2 and you have 2 worms. Okay, like you cut off the head and the worm regrows a head and the other guy regrows a tail and now you have 2. So, and the reason that they can do that is because they have like lots of stem cells, sort of general stem cells throughout the body. Okay, so they are great organisms for studying stem cell driven growth. Okay, that's why they like them. But what Wendy discovered already a few years ago, this was a paper she had, and I was not involved in this. This was before I did these things myself. Somehow she became interested in magnetic fields, and she showed that the magnetic field makes a difference. Okay, to how so the paradigm. And again, this seems mean, I know, but these guys, they are uniquely positioned to handle this kind of abuse, okay? So what she does is she cuts off the head, cuts off the tail, then the thing looks like this, and then it regrows, okay? And you track how fast, basically. How these regions here are called the blastemas, and they can track how fast these blastemas grow. And what they found is that if they apply a magnetic field, it changes. Okay, so for a field of 200 microtesla, it grows more slowly. And then at geomagnetic field, so the Earth's magnetic field in Michigan is 45 microtesla. In Calgary, it's a bit more. This is compared to that. Okay, so if you apply 200, it goes down. If you apply 500 microtesla, it actually goes up. They grow faster. So it's not a trivial kind of effect either. Okay, and this will become more clear in a moment. So that was the initial observation, and then they also showed later on, that was already prompted by discussions with us. Here they had measured sort of reactive oxygen species in general, whereas we said, okay, we are particularly interested in superoxide, which is just O2 minus, right? We had our reasons. And so they showed that it's really the superoxide that is driving it, so the magnetic field effects change the superoxide levels, and then that changes the growth. That was their state of knowledge in 2023. Okay, so it's the superoxide goes down here, and that was very promising for a radical pair explanation, because superoxide is a radical. Okay. Now, we studied this then with our quantum theory models, and so started with a very simple model where one of the partners is flavin, which is the one that, this is basically FAD. I had that on a previous slide on the bird navigation. So this is a molecule that likes to form radicals. And then we said, okay, maybe the other radical is just simply the superoxide radical, because we know that it's somehow involved. That's not necessarily, this is just step 1, most simple, possibility. And but it's good enough to make predictions, okay? And the predictions we made were that, so this is the range that I showed you before, where it was like negative here at 200, positive here at 500, right, effect. And then our model said, well, it should keep going here. This is from this radical pair mechanism, right? It should keep going up here. And then it should also go up here and quite a lot, okay? There should be a large like increase. So it's not, we're not just fitting a straight line to the thing, right, is my point. We are predicting something relatively non-trivial, especially over here in that range. Right here it's just a continuation, you could say, but here it really, you know, that's maybe not, naively you would probably not predict that, right, if you didn't have a model. So we had serious doubts, okay, because this is not what the, so our prediction is for the superoxide. They had some data on how the worms actually grow, and the worms, they don't do that, okay? So we really thought, we encouraged them to do an experiment to actually measure the superoxide, but we fully expected to be wrong, you know, because this was just a simple model. There's many excuses. We were already ready with our excuses why this model is not good, you know. But what happened is that it worked, okay? So they measured the superoxide here, you know, at zero, more or less 0 microtesla field. And it wasn't quite as high as like our, you know, as I showed you before, but it's the highest that they had seen, okay? So it really goes up at 0 field. And it really, you know, this is the negative point, this is the positive point that they had before. It really keeps going, right? Like, so we said basically it should keep going up here, and here it should really go up. And that's exactly what it did, okay? So not like, we didn't have quantitative agreement, but we had excellent qualitative forecasting from our simple model. So that was shocking, okay, honestly. Like we did not think that that would really happen. So because, like I said, the plastima size, which tracks the superoxide here, like here when the superoxide goes down, plastima grows less, goes up, it grows more. It kind of decouples here. What the biologists, they tell us, yeah, that's probably because it gets too much, and then, you know, there's some feedback, and it doesn't shock them at all. Okay, they are used to this kind of nonlinearity. To us, it was really, you know, kind of shocking that, anyway. So I'm not really telling you that I understand how the worms grow, but I think I understand what drives the superoxide levels. Okay, this is not quite the same statement, but like, it looks like the radical pair mechanism really sort of can predict the levels of this particular chemical. So that was a nice success for us. So we are, you know, we have a paper about this. That was example 1. Here comes example 2. Let me keep an eye. I have a clock here. Okay, I have some time. Example 2 has to do with a protein called tubulin, which forms these microtubules in our cells, which are key components of the skeleton somehow of our cells. They are particularly important when cells divide. And I have a personal note there. They recently were very important in my life because unfortunately my wife has cancer. So she had to do chemotherapy. And what the chemotherapy does is it targets those microtubules. Okay, this is what chemotherapy is about. So they are really very important things for like many reasons and also for health purposes. Okay, now it turns out that the process of microtubule formation from this tubulin protein in a jar, so this is an in vitro experiment, is strongly affected by the magnetic field. So in particular, what these people showed, this was a Chinese group already a little while ago, is if you just, again, remove or shield the geomagnetic field, the field of the Earth, you bring it to near 0, then it really makes a big difference. Like, this is a simple illustration, but in their paper, but basically, here the deposit is, this is the polymerized tubulin. If it polymerizes, it falls down. And it's a big difference, right, between this is geomagnetic field, this is the hypomagnetic field. It's a very big difference in this polymerization rate. There's more that goes on in this experiment, but this is just the simple thing I want you to focus on. So there's not like doubt, you know, that that's really true. Okay. Then we wrote a theory paper. That's often how we start, you know. So in 2022, again, my PhD student at the time, Hadi, and just a little team, we wrote a theory paper where we showed that this makes sense from a radical pair mechanism perspective. You know, probably there is a radical pair in there somewhere in the tubulin that affects, you know, how the depolymerization happens, and that's why it's sensitive. So we predicted curves like that just for the rate of microtubule formation. But so one could have tested, you know, just the magnetic field dependence, but we already were thinking about these isotopes too. So we said, let's also play with the isotopes a little bit. And we were at the time thinking about zinc, but it really doesn't matter. You know, any kind of isotope will do, and it will move this curve around. Okay, so generically, when you have a radical pair mechanism and you change the nuclear spin of an atom that's near to your radical pair, that's typically going to make a difference. Okay? And then, so that motivated a colleague who was at the time in Florida, but is now at the University of Waterloo. He's called Travis Craddock. He became excited about this and decided to test it, right? But he had his reasons. He didn't really want to use zinc. He wanted to use magnesium because he knew that the magnesium is important for the microtubules. And anyway, he had his own, like, he is more of a biologist. I mean, he's a pretty physicsy biologist, maybe biophysicist. It's hard to quite, yeah, I'm not quite sure what to call him. But anyway, he does this kind of stuff. So they started doing these experiments, and then actually at some point, he hired my former student, you know, and like now there's this joint paper where they observed the following thing. So we are looking at different isotopes of magnesium. There is actually, so the natural magnesium is mostly magnesium 24. Then there's magnesium 25 and magnesium 26. Magnesium 25 is the only one that has a nuclear spin. It has a nuclear spin of 5.5. And here is basically observing what I showed you on the previous slide, you know, observing the polymerization of this tubulin protein. There is a huge effect when, So there's 3 isotopes, but there's also field on, field off. So they can apply a 3 millitesla, in this case field, or not. Okay? If you apply that field to natural magnesium, not too much happens. You know, there's probably an effect here, but it's not, you know, dramatic. If you apply it to magnesium 26, again, like not too much going on. There's significant error bars, right? But if you do it here to the one that has a nuclear spin, it's like huge. It's staggering, you know, like 5 times, 6 times, something like that. So there's a very large combined magnetic field and isotope effect. Again, bigger than we really wanted. This happens to us a fair amount. You can see that our model, you know, if you look at this here, this is our modeling. So Hadi, with like a lot of searching in a pretty large phase space, he just got this here to the lower edge of that error bar, right? So you can sort of tell that the effect is kind of larger than we really wanted it to be. But we can manage, okay? So maybe there's more going on. Probably there's more going on. But again, it's a very successful prediction. We said there should be an isotope effect in this process, right? And there is an isotope effect in this process. So like now we have this paper here. That's a preprint that came out in the spring. Yes? It's 3 millitesla in this case. So it's a bit stronger than the Earth's magnetic field, right, which is 50 microtesla. But I would still call that weak, yeah. It's still true, for example, that this is the energy of that is much less than the thermal energy. It's still in that range. That changes when you go into the many Tesla. Yeah. Okay, one more example, also very nice, I think. So because the last part of my talk is going to be about photons anyway, which I inherited at the beginning. So this here makes a connection between the spins and the photons. Okay, so ultraweak photon emission. Like all living systems, to my knowledge, emit light at a low level. Okay, so this is called sometimes ultraweak photon emission, sometimes it's called biophotons. They are, it's actually fairly well understood where they come from. Like they come from certain chemical reactions. I'll come to that in a little bit. It's definitely, it's a non-equilibrium effect. Okay, so it's not black body radiation. That's not what we are talking about. So I'm showing this here. Like black body radiation as a function of wavelength has a behavior kind of like that. Okay, so it goes up as you go towards longer wavelengths, lower energies, right? It goes up. But I'm talking about over here, okay, in the visible near infrared. This ultraweak photon emission is much stronger than you see the scale, 10 to the minus 6, 10 to the minus 10, 10 to the minus 14. It's many, many orders of magnitude stronger than black body radiation would be. It only happens because we are not in equilibrium, right? And here is actually, we did this experiment recently, which now just came out in this physical chemistry journal, where we showed that sort of clearly, I think. Maybe for the first time, actually, really clearly. This is an alive mouse, and this is a dead mouse. And you can see that the alive mouse is much brighter. Okay. I want to make the point that the mice were not killed for us, okay? So there was a different study, and like we piggybacked on that study. Mm-hmm. Yes. Yeah, that was an important thing that we made sure. So this is on a stage that is still kept at like 37 degrees, but it's dead, okay? Whereas you can still see a little bit, you know, like maybe we are not totally sure, like maybe it just takes long. This is, I think, has been, you know, this is recently diseased, like an hour or two. So maybe there's still some activity, you know, in certain organs. Sorry? I don't think it's decay yet. It's too soon. I think it may depend on what you call decay also. Anyway, but like definitely these are much brighter. Yes.
For the same period of time, like 2 hours death for all of them?
Yeah, it was always the same protocol, I think. Yeah. Yes. And like he said, they were all at the same temperature. It's not a huge number of samples, right, to be fair. But like I said, this was some cancer study that they were doing in Ottawa, and but we had a joint project, so they had a camera so that they were able to look at them. We can't do mouse experiments in our own lab in Calgary. We would not be allowed. Okay, so there we collaborated with. It was Lana, who was a postdoc, our postdoc, but there in the animal lab, who did these measurements. So these photons are for sure there, and plants have them too. So and for plants, it's particularly well understood where they come from, because people were able to do some genetic sort of knockout things. And the message is, so these are two plants, and this one is the wild type of the plant, and this one is one that doesn't have this enzyme. And this enzyme is essential for lipid peroxidation to happen, which is a certain, important process that can happen. So, and it's almost dark, right, compared to that one, if you look at it in the dark. So these guys, this is a French group, they really kind of proved that the photon emission, at least from the plants, is predominantly due to this process. And what's really nice about that is that this process of lipid peroxidation, it involves radicals again. Okay, so it involves these kinds of R is just let's say a lipid molecule of some kind, but it has these molecules. That's why it's called peroxides. OO, and it's a radical. And it's the process only ends when two of them meet and react. So perfect conditions for the radical pair mechanism, okay, and for magnetic field dependence, therefore. So this is somebody else's paper who they had already thought about this, about the magnetic field part. So they were predicting, they did some radical pair making. This is Daniel Katnick, who has done a lot of work in this area. He's at the University of Exeter in the UK. This was, 2019 already. He predicted that lipid peroxidation should be magnetosensitive, okay, in such a way. But he did not think about these biophotons at the time, okay, because that was just not on his radar, I think. So, but this suggests that if it's true, what the French group says, that ultraweak photon emission is due to lipid peroxidation, and then it's true what he says, that lipid peroxidation is magnetic field dependent, then ultraweak photon emission should be magnetic field dependent, right? And that indeed seems to be the case. So this is pretty preliminary data, so there's, this is still in preparation. But here we are looking at ultraweak photon emission from leaves, because it's really easy for us to do experiments on plants, and particularly on leaves. They still live. You can take them from a plant and they still live for days, so you can easily image them. Their metabolism maybe slows down a little bit, right, as you can see here. So the ultraweak photon emission also goes down. But it turns out that the speed at which it goes down depends on the magnetic field. That's our observation so far. And that makes perfect sense, because we think that this decay is associated with the lipid peroxidation terminating, and we predict that, so it are somehow, it seems, we feel, we are still doing more measurements, and we are working on the modeling and so on, but we feel that this is kind of what we expected. For once, we feel that we even understand the chemistry of it, more or less. This should be this lipid peroxidation termination reaction. So it's really nice, but it's not quite ready yet for our paper. We are working on that. All right. So with that, let me close by talking a little bit about photons. So I already told you that they are there, in the sense that cells emit them. Question is, are they somehow used, right? And in particular, could they be used as signals? Because we know, of course, from the technology side, that light is a very good signal. You can send information with light really well. So it's natural to ask if nature, if our bodies have figured that out too. Now, we know that there are, obviously nature can detect light. That's our eyes, right? So we have receptors in our eyes, and many animals do, that can detect light. They are based on these light-sensitive proteins called opsins. In particular, in our retinas, there's one called rhodopsin, but that is far from the only one. And our eyes also show that it's possible to evolve mechanisms where you can detect even really low light levels, like our individual photoreceptors in our eyes are actually sensitive to single photons. Then they are a bit too noisy, so like at least in our eyes, they are not used like that. There's sort of a coincidence electronics behind that. Only a signal to the brain is only triggered if a few of them respond at the same time. But they actually have single photon sensitivity. So it's a doable thing. There's also papers that show that, I don't know if that's surprising or not, given this context, but there's explicit papers that show that, for example, this is a part of the brain, the hypothalamus. If you stimulate it with violet light, it does something biologically meaningful. This violet light makes a difference to a brain part that's pretty deep inside the brain of that mouse or rat. I forget now. Like it was a mammal. So, this is a fact, I think. This is a very good journal, as you can see, and so on. So this is not some crazy margin, kind of thing. But this was still relatively strong light. So that does not, and this was light that came from the outside, right? This does not show that the photons that are made by neighboring cells do that too, right? This is still an open question. But at the same time, it's not totally crazy. Background light, we have been thinking about this, right? Like there is, of course, background light, because we live in the world, right, and the day is bright and so on, but that can be manageable, especially in the blue, where, for example, for humans, the transmission of our scars is quite low. Like you can send red or near-infrared light through our heads quite easily, fairly easily, but not blue. Okay, this is total cutoff. So you could use blue inside the brain for communication. The ultra-weak photon emission is so weak that it is actually quite hard to characterize really spectrally. I mean, you can do a very broad spectrum, but the resolution so far has been quite poor, like more like tens of nanometers, because you have so low counts. Okay, so and then spectrometers tend to be quite lossy. So that is sort of an open challenge, is to see if there are any narrow lines in there, because it would be good if you want to do communication in a high background light setting. Typically you want to filter somehow. You want to have narrow lines for your receivers and your emitters equally. But we don't even know if there are such narrow lines there, right now. But here is something we did do that makes things easier, which is that this was actually my first real paper in this general area of like quantum biology, or however you want to call it, which was the idea that the axons that we have in our brain, which are usually thought of as electric cables, right? Which they are. I'm not telling you that they are not. So they conduct electrical signals for sure. But they are actually also really well suited to conduct photons. Because it turns out that already, so there's axons, there's two kinds of axons. Some of them are unmyelinated, and there's just the yellow part. It's just a tube like this. But then often there's many myelinated axons that are wrapped in this substance called myelin. And the axon, the yellow thing itself already has a higher refractive index than the surrounding medium, which is basically water. But the myelin has an even higher one. Okay, so the myelin should be, and that's what you need for light guidance. So especially the myelinated axons should be really good light conductors. And they seem to be. I guess I should speed up. So at the time this was a theory paper, like we often start, right? We tried to include all kinds of imperfections that are of course there in the real biological system. Here's some examples. We were trying to see, because the myelin sheath has these parts where it's interrupted. They are called Rabier nodes. It has funny shapes and stuff, but it seems like it should still work. We have also looked at whether polarization could be transmitted, and it seems that could actually also be the case with like decent fidelity. There is also some experimental evidence, that these myelinated axons really guide the light. For example, here we're looking at a slice of brain, and these are the axons, and you can see, so it's illuminated from the bottom. I think we're looking at it. You can see that these are bright, right? So this is the myelin. So the prediction would be that, or the explanation would be that this is where the light is guided by the myelin. But we do want to do more, and we are trying to do more experiments where you could really sort of characterize how well this like light guidance is working, what's really the transmission, but this is really fiendishly hard to do actually, but we are trying. But there's at least indirect evidence that it's true. Okay, so now, do I have to close sharp at like 12 or no? Okay, maybe I'll spend like a few minutes on this. So this is, a big question, obviously. So could the brain really use it? Like I've been arguing that biology could use it somehow, but I am interested in this question of whether the brain could be a quantum computer of some kind, right? I want to make the point right away that, we know, of course, that there is a lot of classical information processing that's going on in the brain. that's also true in a quantum computer, right? Like if you look at a quantum computer right now, there's a lot of classical information processing that's going on, no question. There's just a little core somewhere that's actually quantum. So I think that's how it would be in the brain. If it was the case at all, there would just be some sort of quantum core hidden in there somehow, or a quantum layer, something like that, right? So it wouldn't be for all the tasks or all the parts. Now, one possibility that's very intriguing is that it could have to do something with consciousness. That's something that motivates me. So consciousness has this kind of holistic character, which is a problem from the point of view of classical physics or philosophy. People are talking about the binding problem of consciousness. How does it arise? How does our unified experience arise out of all these molecules that we have sloshing around in our heads, right? So this is a notorious problem, and it seems like quantum entanglement could be a solution. Quantum entanglement is a holistic thing, and it's very tempting, at least for me. That's a very tempting idea. I have argued that the basic ingredients are there, right? We could maybe build, I showed you that there's pretty clearly there's spin effects. We also have photons. We could probably maybe build something out of these spins and these photons, a quantum information, quantum network of some kind, right? Distributed quantum computer or something. But I'm not telling you that that's true, okay? I'm just saying maybe. I think it's an interesting question, but of course we need a lot more work. We don't even know yet that the photons are signals at all. We don't really have the specific spin-photon interfaces that you would need, okay? So this is a fascinating question. It's not a claim, just to be clear, okay? But I do think the claim is just that it's a fascinating question. Then I'm almost done. So then that leads to another line of inquiry, which is I've been talking about the hardware side of it. What would be the software side? Suppose that the brain has these quantum ingredients. How really would it use them? What kind of quantum computing or quantum learning are we talking about? That is far from obvious. Would there really be C-naught gates, and stuff like that, or is there something else? More like a quantum neural network. So I think something like a quantum neural network is much more likely, right? Because that's the architecture that we have. So we have recently been starting to learn and look into these things. We have done some work on something that's called quantum reservoir computing, which is a very, very simple thing, really. I won't go into the details right now. But we are now trying to build on much more sophisticated things, which is approaches to AI that are already really inspired by pretty sophisticated theories of consciousness, which are called workspace-based architectures, where the idea is that you have sort of a modular processor, and then there is sort of a global shared workspace between these different modules, and that's what is tentatively identified with our consciousness. So there's an idea, sort of a neuroscience model underlying this, and then the idea would be that maybe it's that coordinating module at the center, that would be quantum. Okay, so the other modules would be classical, but then this module at the center, this workspace, which is where consciousness lives, would be the thing that's quantum. I think that's the hypothesis that's sort of driving us now. So we are starting to work on these quantum workspace-based architectures. But the classical workspace-based stuff, that is it's not the mainstream in AI, but very serious AI people like Yoshua Benjo in Montreal are working on that. Okay, so it's not some crazy thing. But the quantum part we want to add. All right, so here are my conclusions. So spin-related quantum effects actually seem to be common in biology. That is my claim. We have now made many proposals, but now we have made several successful predictions. And I showed you, right? I showed you the superoxide in the worms, the microtubules in a jar, and then the photons from the leaves, right? And in all of those cases, it seems to work, even if often we don't know the chemistry, the exact chemistry. Then photonic signals in biology are at least possible. Certainly the photons are there, and it looks like we have waveguides. That would, of course, help immensely, right? If you could guide the photons, it would be much more specific and targeted. And then quantum information processing in the brain remains highly speculative, but there's various reasons to think why it would be attractive, and we are starting to think about how precisely it could be used, also from a software perspective, I guess. And with that, I thank you, and I thank my various colleagues and collaborators who have joined me in this adventure. Thank you.
Okay, thank you very much. We have time for questions. Just please wait for my microphone to hand you if. Yes, okay.
One question is actually broader. Do you know of other works tackling maybe other theoretical explanation different from these radical pairing mechanisms for other quantum-based phenomena in biological systems?
These were two questions, I think, right? So I'm not sure I understood the first one correctly, but so are you saying for these magnetic field-based phenomena, for example, could there be another quantum explanation or so? Yeah, I don't think we can exclude that, because it's just the radical pair mechanism is a fairly well-established thing, so it was natural to start looking there. And it seems that we can make successful predictions based on it. So there is not a strong incentive, to look for dramatically different things right now for us. But could there be? Yeah, we haven't even scratched the surface. Some people are looking, for example, Daniel Katnick, who I mentioned, he's very interested in radical triads. If you have a third radical joining, the first two, then other things can happen and so on. So I'm sure there's more to be seen there. Then I think you were also asking, are there other phenomena completely, of quantum biology? People have certainly looked. I mentioned briefly photosynthesis, where for a while people were very excited about these coherences that they thought they saw in the movement of the charges. But there they seem to now have concluded that that's not really the case. There isn't really coherence there, which makes sense to me, like I said, because charge movement is not something I would really expect to be coherent other than on really, really short timescales. So to me, I think if I knew of another really promising area, I would try to work on it too. So I showed you the ones that seem most promising to me, which are the spin phenomena and the photon ones. But not everybody might necessarily agree with what I'm saying. Right?
So I was wondering, long time people, there are reports of people claiming that there are negative health implications of quote-unquote electrosmog and stuff like that, complaining to live under high under electric lines and stuff like this. Do you think these claims are dismissed too easily and should be re-evaluated in the face of your research?
I think at least in some cases, yeah, I think so. I think we have to consider, because we see that even quite weak fields, right? And the biologists know that, right? There is a lot of papers where you can see that even pretty weak fields, these are fridge magnet fields that we are talking about here, right? Have pretty dramatic effects. Yeah, that doesn't mean that, because at the same time, these effects from things like power lines, they do decay with distance pretty fast, right? Often you have 1 over r squared at least, sometimes more. So I don't know that every concern that people have ever raised is necessarily justified, right? But like, if your fields are in that range, you know, of like microtesla to millitesla range, you gotta like really take that seriously, especially if you have long exposure times. Yeah. If you just walk by it, it probably doesn't do anything. But if you sit in it for a week, yeah.
Because I'm following this up because, you know, recently, also during the pandemic, there were all these concerns about 5G basically, you know, having detrimental health effects. And I mean, back in the days, everybody was like, yeah.
So I'm more worried. Okay, no, it's a complicated topic, but people have also observed effects from gigahertz fields. Those may be just as real, but they are probably not due to the radical pair mechanism. That's what I will say. So we actually have a paper about that. That would probably be, so that there the predictions for the radical pair mechanisms would be that these effects are really small. But so they are probably due to, if they are real, which I'm not, you know, necessarily doubting, they are probably due to the electric component of those fields, okay, like in the gigahertz range. If you go down in frequency, like megahertz range, this becomes important. But what I showed you mostly was really constant ones. So I'm more worried about, you know, and this has actually changed with the generations, I think, I read. I'm more worried about just, you know, there's currents in your phone, right? So there's a constant magnetic field that this thing makes. It is now much less than it was in the earlier generations of the phones. Should you really always have it in your pocket, you know, or like, probably you shouldn't carry it on your head at least, kind of thing, right? But measure the fields, you know. I think people should really like think about if you have a device that you're going to be close to and it's right on you, like for the whole time, you know, then let's figure out how large those fields are. And if they are small enough, they are fine, right? Because we are in the Earth's magnetic field all the time. Yeah, we are probably optimized for that.
Thank you for this great overview. I really enjoyed that. And I have a very question about, did you investigate, or are there enough studies about how does the fundamentals work of the radical pair mechanism if you put it in a magnetic field? I mean, you have two spins, sorry to help me because I forgot how it worked. You have two spins that are just opposite sides, but the orientation is not known, so you put it in a magnetic field, right? And they start to precess, but you in addition need some kind of decay to get it back oriented, or like a Zeno effect to keep it in the singlet state, right? So it's...
So you'll have you can have those things, but the basic principle of it is that if you have the two electron spins, and then you have at least one nuclear spin, that at least one of them is coupled to, then you are already have broken this symmetry, and you are going to oscillate between singlet and other states, you know, singlet and triplet states. That's sort of the core. Then, yes, then, you know, of course, you have spin decoherence, and then you have the chemical reaction itself, you know, and that's a decay process and so on. So it is, in fact, an open system, right? But like just the sheer oscillation, which is really the core of it, the only ingredients that you really need is two electron spins, one nuclear spin. That is coupled to one of the two electrons and not the other one.
Plus the nuclear spin.
Two electron spins, one nuclear spin. Yeah, that's the core part that you need.
Okay, very nice overview, thanks. Different question, do you have, I mean, your main focus is biology here, so do you have some more examples of these quantum effects where there's a known biological function? I mean, most of your examples were showing an effect on biological systems, sometimes quite strong, but not necessarily in the sense that it has some useful biological function. Of course, the prime example was the magnetic field with a bird migration. That's a very clear example. Do you have some other examples in that direction of other?
No, I don't think I do. No, I don't know if it has been. Yeah, like you said, you know, there's the distinction between sort of sensitivity, right? Magnetic field sensitivity, which seems to be extremely common, and then magnetic field sensing, which is sort of intentional, right, or seemingly intentional by the biology. And there, the only example I know that's really sort of established is the navigation. But I think that's a very interesting question to see. I have heard some people speculate about other ideas, but I don't think there's anything clear on that. If you have any ideas how to study that, that would be an interesting topic, I think.
Thank you.
Thank you for the talk. My question is, do you think the information transmission across neurons also carried out by biophotons, not just by action potential progression, which is much more slower? Because we know that in myelinated axons, it is much more faster than unmyelinated ones. Do you think biophotons can explain this?
I don't. So I think one can understand the fact that the myelinated axons speed up the electrical signals. One can understand that with conventional classical physics of how the currents flow and so on. But I am suggesting, yes, that maybe there could be an additional channel, right? That would be this like photonic channel, which could have all kinds of advantages. So one is like you said, it could be faster, although you also have to think about the speed of the sender and the receiver. So that's a bit more subtle. It takes you some time to detect the photon, and for example, our eyes are not very fast. So that's a caveat on that one. It could be another one that I didn't mention is it can be bidirectional. So whereas the electrical signals in the brain, typically they always go in one direction along an axon, the photons wouldn't necessarily have that constraint. And we actually have a paper sort of proposing that could be one way in which backpropagation could work in the brain. There's this algorithm that's very popular in AI that's called backpropagation, and it's a bit of a mystery. It's really useful in technological AI. It's not clear that it could be implemented in the brain or exactly how that would work, because you can't easily go backwards. So people have various ideas about that, and one of those ideas would be that maybe it's the photons going backwards. The reason I am really interested in is that it could be quantum. Because the photons, unlike the electrical signals, which are not suitable for quantum communication, the photons would be. So that is my personal motivation, but it's not the only thing.
Okay, I would suggest that we slowly close, but so the last question, please.
Yes, I was wondering, now you have isolated, for example, this process in plants that produces these biophotons that is somehow very sensitive to very small magnetic fields. Is there an avenue where you could isolate just the chemistry, right, of it for purpose of detection, sensors, technological applications? And is there also some kind of inverse, like could you use this process inverted, like start with the photons, have a very sensitive photon detector that can produce, detect efficiently very small photon fluxes?
Right. So sort of the connection to technology of all these things. So there are actually quite a few groups who are interested in using molecules for quantum technology more broadly. And then some of those groups are looking at radicals, like usually stable radicals. And then similar physics intervenes to here. So that's something I'm aware of. Sensing is also very natural. I'm a little bit less familiar, but it's a very natural idea. Could you build an in vitro sensor, using this? Because it works. They showed with cryptochrome, for example, right? In vitro, you see the magnetic field sensitivity. With FAD, similarly, is that there's more and more. Recently, a few groups showed ODMR, with some proteins and stuff. So yes, people are thinking about that. Light detection, I have been thinking myself, actually, can we build a room temperature photodetector, for example, using these principles. I was excited for a while. I got discouraged by the slowness of it all, but maybe there could still be a niche, where that would be really useful. you know, because our eyes, it does take milliseconds, right? You would be limited to pretty low rates if you follow. Because the principle how it works is you have, you know, the photon excites, of course it can only excite one molecule, right? Now you have one molecule. Now you need to amplify that somehow. So the way that biology does that is that this molecule is a catalyst, and it's going to catalyze a bunch of other reactions, maybe 1,000 of them. And then actually what happens in our eyes is that there's another stage where now these guys that were catalyzed, they are themselves catalysts, and they catalyze another 1,000 reactions, each of them. Now you have 1,000,000 molecules, and now you're talking. You know, now this is something that you can actually like sense at the level of a cell or something. But that takes time, right? So all these reactions need to happen, and then they need to happen again. And so that's why it's slow. If you have an idea to speed that up, you know, maybe you put it in a tiny little volume, you know, where they all meet a lot of times, and then yeah, who knows? I think that's an interesting topic too.
Okay, great. Thank you very much, Christoph. So let's thank